Hi, everybody. This is the continuation. This is part two of the review for exam one. I just sent the first one, depending on whatever order you're seeing these in. And so the next set has to do with the proteins we discussed. So this is the one of the aspects of the cell that you should be familiar with. The other one is those cell adhesion molecules, and I'll talk about that in a second. Effectively, as I said in class, you're going to see basically the answer. It's the same thing, the answer key. I'll give it to you. Enzymatic proteins, cell-to-cell recognition proteins, attachment proteins, channel proteins, signaling proteins. That's the answer key. You figure out from that key which applies to the phrases I will give you. If I'm talking about any of those things, if I'm talking about something like the way insulin works, effectively that would be something that would have to do with the way things are signaled or signal transduction, as we covered in class. Recognition, we talked about with those short chains of carbohydrates, etc. Enzymes, you remember the location I told you about. If you don't, go back and listen to recordings. That's what I can tell you from this. Okay, all of those are parts of that you need to be familiar with. Okay, so I know what the questions are pretty much, or how they're going to be phrased. So when I'm in lecture, listen to my points of emphasis. If I go over something several times, most likely it's going to be on an exam. So you need to understand the distinction between integral and peripheral proteins, exactly what that means. That'll be fine. Okay. Those intracellular junctions, all those cell adhesion molecules, those terms are interchangeable. Which one is the strongest? Which one reflects being watertight? Which one is able to allow either chemical or charged particle signals or somehow cells-to-cell communications? Those are all part of what you need to be familiar with. So that's a series of questions that regard those. Okay. And that really was the last part of that cell chapter that we're going to do. The rest of the cell chapter is what we're covering in class at present, and that's where we start with that thing called movement across membranes. Understand the composition of these cell adhesion molecules. We went over several of the proteins and several of the materials that were involved, and I think you need to be familiar with that as well. As we go back to when we started the class, the very first lecture we did the organizational hierarchy table. That's something that you need to be familiar with. What makes what, and the concept of emergent properties. Remember, we talked about that when we had atoms. Basically, when we put two atoms together in some way, shape, or form, now we have a molecule. Molecule is an emergent property, whether it turns into oxygen gas, or it turns into salt, or it turns into glucose, being different than just the pure atoms themselves. As we move on from that, we build large molecules, and really the big molecules start to really give us properties, whether they're proteins and all the roles we've gone over, or DNA and RNA. Those are macromolecules. And so the emergent property, when we put those together, and typically when we encase them in yet another large molecule called a membrane, now we have something like an organelle, and the organelle begins to have significant properties like a mitochondria being able to produce energy, or a ribosome being able to assemble proteins. And then as we move farther with that, we take all of those together and encase those in a larger membrane. Now we have a cell. And that is certainly the first unit that really meets the test of all living things. Because at that point, it can, even though we are multicellular, but biologically there's unicellular, both unicellular animals and unicellular plant life. And as you'll see, bacteria are at a whole different dimension down the road. When you learn about that in micro, they're just made out of cells. So at that point, the cell means, even whether it's ours or not, they can reproduce, they have DNA and RNA, they have the ability to respond to stimuli, all of those types of things. They do energy transformations in order to make things occur within the cell, all the things that meet the test of life. So be very, very familiar with that. And then what happens after that? We have the same cells, we have a tissue. If we have two or more types of tissues, at a minimum, we have an organ. If we have organs that function similarly, we have an organ system, and then we have us, which is an organism. So all of those things are part of it. Again, be familiar with the norms that we discussed. Remember, the language of science is not Fahrenheit, it's Celsius. The language of science is metric. Okay? It's not, we don't talk about pounds, we talk about grams and kilograms. So understand that those are things that you should be familiar with. Okay, if you are, and you're not in this particular thing, to do a calculation, then I would provide you with effectively the what to what, like how many ounces in a pound type of thing. Even though you're all familiar with that, but we're not doing a calculation in this particular exam. Remember how the role water plays, just how much of it we're made out of, as we discussed in lecture. And then certainly homeostasis, such an important core concept. The state of internal balance is basically continuously adjusted by something we call negative feedback. If we move away from that internal state of balance a little bit, mechanisms go into action to bring us back, okay, to reduce the difference, so to speak. That's why I call it negative feedback. The way we go away from our set point, we reduce the absolute value of that and bring it back down to our set point. Nearly all the feedback mechanisms are negative. The two major ones for now that we studied that are positive, and basically they take the event and bring it to its conclusion faster, as a much better way to address the problem rather slowly, because it's a significant problem, like the birth process with oxytocin, or was made is called pitocin, to basically make the contraction harder and expel the baby. So it's amplifying or cascading response, like blood clotting. That's what platelets do. They release chemicals that don't stop being made until such a time as the blood is completely clotted. So know those distinctions. Everything else, whether it involves calcium, antidiuretic hormone, glucose, temperature changes, all of those things, okay, are controlled effectively by negative feedback. And I'm just a typo, but I'll talk about that when we get to class. I beg your pardon, we do the test. So all of those things I've covered just now are so important. Okay, and again, remember, just underscoring, remember those emergent properties, all that stuff I just told you about homeostasis, that's effectively all covered. We'll talk a little bit about the basic chemistry components. You have, remember, this is stuff that you should have had ages ago: protons, neutrons, and electrons, which ones have charge, which ones have mass, which ones don't have mass or effectively have negligible mass, which ones have mass but no charge, etc., etc. You know all those things. Review it, okay? And again, just to underscore, and I mentioned it before, those number of unpaired electrons in the outermost orbital, the whole idea of valence, that's where the chemical properties come from. That's the valence shell, another name for it. All of those are part of it. And so then, and the very last part that we need to come to, it has to do with the kind of bonds. Remember, and effectively, that's when we start talking about water and we address the rest of these things. And so I'll give it a little rest now, and I'll complete this process very, very shortly. And so this will be number 2. It'll be our third one. So you can review the areas that you think you need help with, or you're a little confused for separately. And each of these will be labeled as review recording 1, 2, and then lastly 3. Thank you.